Ramon y Cajal did for the rest of his career, which is use that stain, look at different slices of brain tissue and remark on how wonderful and complex the architecture of the brain is. One of his comments is that the brain is an impenetrable jungle, this is towards the end of his career, where many investigators have lost themselves. So fast forward to today, it's now possible to stain all of the neurons in the brain and to really stand back and look at its full complexity. I teach the physics of measurement at Caltech, and it's hard to even know where you would begin with something so complex. You see a region of the hippocampus here where the neuron bodies, the soma, they're about 15 microns in cross-section, are densely packed, and spreading out from them are maybe 1,000 to 10,000 dendrites, we call them processes, the cabling from the individual neurons, they're reaching out to connect to other neurons. In terms of its complexity, lots of smart people have said this is perhaps the most complex organ in the universe. Even Roger Penrose saying that the galaxy is just an inert lump compared to the brain. Yet, it's interesting to compare this complexity to the complexity of a supercomputer. The human brain has about 100 billion neurons, and each of those has 1,000 to 10,000 synapses. And that number, 10 to the 15, is typical number today of transistors in a supercomputer. So engineering paradigms exist that are at this level of complexity. And this was what we pitched back in 2011 and 2012 to the White House Office of Science and Technology Policy, that we could use advances in neurosciences plus silicon large-scale integration to build a new class of tools to massively interrogate the brain. And a couple of years later, Obama announced the Brain Initiative, and the nation has begun to engage in actually thinking about upscaling the technology. Let's talk about resolving brain activity. It seems like every major development in biology is predicated on a technological advance. And you might not recognize this as a technological advance that was really important for neuroscience, but indeed it was. Lee DeForest and a host of other people, he gets most of the credit in history for whatever reason, developed the vacuum tube triode. And this was mass produced in the early part of the 1900s and made cheap enough so that amateurs could actually get their hands on it, experimenters, and build circuits. And one of these experimenters was the to-be-called in future Lord Adrian, Edward Adrian, who built a circuit for amplifying small voltages, and he plunged some electrodes into neural tissue and recorded for the first time with this sensitive amplification this quasi-digital form of communication to and from neurons, the spiking of neurons. We can go forward and draw an imperfect analogy between electrical systems and neural systems. We think of computer processors as being architectures of computational elements, and this is what we're thinking of in terms of brain circuits. I show here a picture of some cortical columns, and these are going to figure later into the story that I tell you. These computer processors are built from individual elements, just like brain circuits are. The individual elements in computer processors are, of course, the electrical logic gates. And in brains, we think of the building blocks as being the many different types of neurons. And this shows 6 layers of a mouse cortex and different types of neurons that can be found in the different layers. And then finally, down at the elemental level, where the bits fly, the logic gates have discrete electrical levels that we represent Boolean logic with. And is there a similar sort of language that the brain talks in with these quasi-digital electrical spikes, these action potentials that the neurons have? We know how to approach the measurement problem for electrical circuits. If you have a computer processor, you put a multiplicity of electrical probes on, and you generate the cyan traces that I show here, the spatiotemporal firings, if you will, of the logic gates in the brain between these different Boolean states, 0 and 1. We can do the same thing with extracellular electrophysiology. We can make microscale probes around the same size as a neural body, then the cell itself, the soma, about 15 or 20 microns in cross-section. We can make probes around the same size that are studded with electrodes, and we can measure in the extracellular environment weak potentials, like Lord Adrian did, of the spiking of neurons, and we can do this in a massively multiplexed way. So I'm going to talk about the future of upscaling this kind of technology using this amazing paradigm for manufacturing we have from silicon very large scale integration, and what the future looks like charting a path forward using this paradigm. Of course, this paradigm is driven, or follows, if you will, Moore's law, which is an exponential rise in the number of transistors. There's many different metrics for Moore's law, but as originally stated, it was basically every 18 months a doubling of the number of transistors that could be crammed onto a chip, and this has proceeded essentially unmitigated for the last 50 years. And an important juncture was crossed around 2010 when it became possible for us to put 1 billion transistors per chip. The consequences of this, of course, are that processing speeds have continued to trend faster and faster. The unit of processing speed is the floating point operation per second, and you can see these orders of magnitude increases that have happened since the silicon integrated circuit has become mass produced. This shows the slowest and the fastest of the top 500 supercomputers in the world. If you sum their activity as if they were harnessed coherently to work on a single problem, you can see that we're almost up at exascale computing, 10 to the 18 floating point operations per second. It's said that by 2020, the individual supercomputers will be at exascale computing. We can ask the question, we know about neurons firing, we know how many neurons are in different brains. If we assume that the average firing rate can be multiplied by the number of neurons in the brain to give us some sort of ensemble average brain operations per second, let's call those BOPs, give them a fanciful name here. What is the rate at which brains fire? So a mouse brain would be on average about 75 megabops, monkey brain 6 gigabops, and a human brain with 100 billion neurons at basically 1 hertz firing rate is 1/10 of a terabop. And if you compare that to flops, you see that the brain is firing 10 to the 7 times slower. Yet, we are conscious and supercomputers are not. So clearly, the basis set for computation in the brain can't just be spiking neurons. There's got to be more. So what are the unobserved domains? So I'm going to address different signal domains, if you will, in the brain and describe how we are pushing forward to actually measure a multiplicity of different domains. But you probably know of a fanciful proposition, in fact it's in your own magazine, reported on, by Matthew Fisher about whether phosphorus spins in the brain actually are coherent enough to be a quantum computer. And I have no idea, but I do believe there are a multiplicity of different domains that need to be invoked and clearly need to be necessary to generate activity, circuit activity in the brain. In the next part of the talk, I want to describe what we've done to upscale extracellular electrophysiology. And where I'm going to get to in this description is that we can't get to where we want to go just by extracellular electrophysiology. And then in the part of the talk following that, I will propose a new paradigm, which we've been following with Brain Initiative funding to go beyond what can be done with extracellular electrophysiology. So let me start with that. So even when Lord Adrian gave his Nobel speech 6 years later after seeing spiking for the first time, he already understood that individual neurons weren't going to tell us the story of how brains compute. We needed to do this, he said, in large numbers and in a way where we could find out the interactions of neurons. Now, despite that injunction, if you will, or that impetus that he gave us, we have made remarkably slow, if not pathetic, progress towards that goal. Now you see the reason why I put the axis on here is I wanted to compare where we are with extracellular electrophysiology in behaving animals vis-a-vis total brain activity. And you see the evolution of technology over the last 50 years since the development of whole cell recording in neurons. And you see that if we extrapolate from this sort of, takes us up to 2015, 500 neurons, and I'll show you we're upscaling from there. It would take another 85 more years, about the same time since Lord Adrian measured those first spikes, before we're going to even be able to measure the full activity of the lowly mouse. Not to say anything about the monkey or the human. So we need a different paradigm. We need a different slope. We'd like to see this in our lifetime, so how are we going to do that? Here is our latest data, published last month. And it looks like it follows the same line, but hold on a second there, because there is a different slope, as I'll show you. The different slope that we can project is because as I mentioned, we're doing this using the billion-dollar scale resources that semiconductor electronic has that allows us to build millions of devices on chips. And I've formed with colleagues in Grenoble that has a billion-dollar research foundry in 2006, the Alliance for Nanosystems, very large-scale integration to pursue these kinds of ideas that are tangential or off-axis from using VLSI for computation. So over the past 10 years or so at Caltech, we've developed at the Nanoscience Institute university-style sort of prototypes for these probes, but obviously it's important to deliver these in boxes to neuroscience laboratories worldwide. And that's what the power of large-scale integration and production does. These are done on 200 millimeter or 8-inch wafers. Here's an individual probe. And if you look at the business ends, they're just what I said, these narrow probes that are about the same size as a cell body that can be sparsely or densely studded with electrodes as you see fit. And we just got a grant from the NIH to massively disseminate these to the neuroscience community over the next 3 years, so we'll be pursuing that. Now, it has to be said that often, and I've fallen into this trap, nanoscientists get enamored with the widgets, the devices, the nano things, and forget that they have to be used in the context of a whole system. And the system is the entity, the holistic entity, not the individual, front end that might be some cool nanoprobe. Here, what's important in the context of the system is not only the nanoprobes, but how you connect them in a way that can be non-perturbative to the brain, where they can float in the brain, where you have front-end electronics that's sufficiently low power that the brain is not heated more than or less than a tenth of a degree, because neuromodulation thresholds change. And so every piece of the system from front to back has to be thought of as an entity that needs to be engineered. And so we've done that, and several other groups around the world has done that as well. But just to show you what the state of the art is, these are layered systems. When you make large-scale integration, everything's obviously flat. You stack things to get 3 dimensions, so we can stack these to make a grid of these electrodes. These have been then implanted into the mouse brain, into the hippocampus, a region that controls sleep and memory. And you can see certain regions. Remember those dense bands of cells that I showed you in the first slide? Certain of those regions, you can see those spikes, aka, also seen by Lord Adrian, and so on. So just to summarize this part, we're still making pathetically slow progress compared to the problem at hand. This recently published work that we worked on for several years has gotten up to 1,000 channels out of 100 billion channels in the human brain, or 75 million neurons in the mouse brain. But this is all predicated on deterministic technology for large-scale integration. And so we know that exactly the steps. It's just engineering steps now and money to actually get up to recording simultaneously 100,000 channels in the brain and disseminating these to the neuroscience community. But that's still a drop in the bucket. I mean, it will be fantastic to be able to do this, to have this multiplicity for interrogating brain circuits, but you can say, is there a path beyond? And so I want to now step back for a moment and think about the physics or the physical chemistry of extracellular electrophysiology and talk about the limitations. And these are best framed in the context of a specific experiment, okay? And the specific experiment addresses some of these early pushbacks that we had from the neuroscience community when we proposed the Brain Initiative, and that is what constitutes a brain circuit. In cortex of the mouse and in us, there are certain structural regions, columnar regions, that have grown up from a single neuroprogenitor cell, a single neural stem cell. And these regions, cortical columns in the mouse, typically are about a millimeter cubed volume and 100,000 neurons. They have many more connections internally than they do outside by an order of magnitude or more. This sounds like a computational element in the brain. And this is what we're focusing on. We and numerous other people are focusing on as an initial place to look for functional motifs, computational motifs in the brain that are conserved across species. My friend Rob Phillips likes to talk about hydrogen atoms, equivalence in biology, looking for things that are fundamental, that give us intuitive understanding, and that can be generic about how the brain computes. And sadly, there are very few such paradigms that exist for how brain circuits work. So the target would be to measure 100,000 neurons in a millimeter cubed. Can we do that with extracellular electrophysiology? So let's talk about electrophysiology for a moment. This shows an extracellular electrode. It's actually 4 microwires that are twisted together, cross-sectional dimensions of about 30, 35 microns. You can implant these slowly in the brain and record individual neurons firing for months at end. So we know we can actually implant these in a non-perturbative way into the brain. And right next to it is a little micropipette filled with an ionic solution where you actually puncture the cell membrane and are enabled to do whole cell recording with the full action potential developed across the cell membrane. So here's what the beautiful signal to noise is that you get for the intracellular electrodes. But the extracellular electrodes, you get this horrible, crappy signal to noise here, which if you do signal processing and filtering, you can see that there's a replica of the spikes, but it's really a small residual replica. What you had to forfeit in order to allow yourself the ease of not positioning the pipette exactly on the cell. I mean, experts can only do 12 cells at a time. That's sort of the record. Whereas I just showed you that with extracellular electrodes, we can do thousands. The price you pay, though, is 3 orders of magnitude, typically drop in signal level. And there's some sort of recording range which begins to decay, and that's what I want to describe now. So this task of recording from 100,000 neurons in a millimeter cubed really would be made easy if it's possible to multiplex many neurons. My pointer's not working. Many neurons are talking to the same microwire electrode. And that, of course, happens. You see multiplicity of different signals that are being generated at the same time. And the technique of spike sorting allows you to use multiple channels to do waveform recognition and time correlation to actually do a signal recognition and to separate these out in a data hyperspace where you have individual units, which you then assign to individual neurons in the brain, okay? And so at the end, even though neurons are simultaneously firing, this multi-site electrode paradigm allows you to measure a multiplicity of neurons. Now, this would be great if it's possible to measure many neurons with a single recording element, right? If we have to put in, well, I'm getting ahead of myself. So let's ask the question, over what length scale is this protocol effective? My friend and colleague and collaborator, Yuri Bazaki, back more than 10 years ago, published a paper where he described sort of the range of these tetrodes and showed that within about a 50 micron radius, you actually have good enough signal-to-noise ratio. Remember, the signals are decaying as you move your electrode away from the neuron. I didn't actually say why can you actually detect outside the cell. Well, when the cell fires, ion channels open up, ion currents flow. When the ion currents flow, you actually have potentials outside, extracellular potentials that you can measure. But obviously, from screening, decay of the ion currents, and the rising background of all the other neurons that are simultaneously firing, this is going to decay into the noise as you move further and further away. And what Bazaki showed is that empirically, things that are within about 50 microns of the tetrode can be recorded, but outside that range become unresolved, and you're unable to do the spike sorting. And what this leads to is that when you have these multi-site electrodes, such as the ones we're building, even though on occasion you have hero electrodes that allow you to record simultaneously, let's say from 5 or 6 neurons, the average is, some don't record from any, and the average ends up being empirically 1 or 1.5 units per electrode. So going back, I won't go through all these numbers, but going back to our problem at hand, you can ask the question, how much tissue, how many electrodes am I going to have to put in? How many spikes? How much tissue am I going to displace in order to record from 100,000 neurons? And the bottom line is, you just tear up the neural tissue too much, even with what I can project for the next 5 or 10 years, the advances we can make. So a different paradigm is required, one that allows us to do more than 1 or 1.5 units per electrode. We need to increase our multiplexing per recording site to get down to levels that are perceived as essential, like 1% tissue exclusion or less. We need to increase our recording site multiplicity up to about 40 units per site. So I showed you where we're going with this grant that I have for dissemination. We'll get up to 100,000 neurons. It's on a different slope, as I said. But there's kind of an end of the road there for extracellular electrophysiology, at least if you're trying to record densely from specific regions of the brain. We need a different technique. And that technique, which we're pursuing and funded to pursue now, is integrated neurophotonics. So I want to talk to you now about photonic nanoprobes. So why am I talking about photonics? I mentioned that there's got to be other domains of brain activity in which information is encoded, and we should be measuring all these, as many of these simultaneously as we can, but obviously none of these are in the optical domain. To use the optical domain, we have to transduce, is the rubric, brain signals into the optical domain. And we do that with reporters, or in the case of trying to stimulate the brain, actuators or effectors. For measuring action potentials, these voltage spikings of neurons, we need a voltage reporter, and this can either be a nanoparticle reporter, or more prevalent are genetically induced reporters, so-called optogenetics. And then once we actually have these molecular scale reporters in close proximity to the neurons, their optical susceptibility will be temporally modulated by the local activity of the neuron. That's the information we want to get our hands on. And we can get our hands on it by essentially interrogating them with a photonic system, okay? Optical interrogation. We send in excitatory photons to induce fluorescence of these reporters, and their fluorescent photons that are coming out will be encoded with the information we seek. You've probably heard of various forms of light sheet microscopy that have really massively upscaled the ability to record simultaneous activity in various organisms. Here's some work from Janelia. 30,000 neurons, much more than our 1,000, but there is a price that has to be paid, and that is you've got to be able to get the photons in. This has worked well with zebrafish larvae, little, essentially worm-like objects, larvae of fish that are about 300 microns in cross-section. But if you go to other organisms that are thicker, you're out of luck. And you're out of luck because of the scattering of light in brain tissue. If you look as a function of wavelength, from the visible to the infrared, at the decay length, this is basically the, if you will, the penetration depth into brain tissue. Large decay length is good, short is bad. For absorption of the photons, we're actually really good in the visible range. Centimeters light can actually penetrate without absorption in tissue. But where you get killed is, in fact, that the brain scatters like milk, if you will. It's called Mie scattering. And the sort of geometric mean of the two, which is the important metric for optical imaging, the optical attenuation length, is down on the scale of 100 to 200 microns when we get in the visible range. And so what that means is, if you think about this mouse brain that I've been showing you multiple times, you can image with free space with these wonderful techniques that allow massive multiplicity in the shallow regions of cortex, but not anywhere deep in the brain. So our paradigm is to use the best of this technology, but in fact to implant all of the elements of an optical imaging system deep within the brain. We call these E-pixels and D-pixels, emitter pixels and detector pixels, using the same sort of silicon large-scale integration technology where we can put these on microscale needles that the brain tolerates if they're implanted properly. And we can orchestrate a deterministic array of these emitter and detector pixels, the components of a complete imaging system. So just to continue with the cartoons before I show you the technology, here's imagining taking one of these multi-probe systems, implanting it into the hippocampus, and basically each of these little microprobes, these little shanks that are implanted into the brain, have interrogating light beams. They cause optically labeled with these molecular reporters that I described, neurons, cause them to fluoresce, and that information will be encoded with the activity, the local activity of the brain. So these probes have photonic waveguides, emitter pixels, and single photon avalanche diodes to actually count the photons that are coming off. Now, this turns out to be somewhat similar to the spike sorting regime that I described earlier. We're not actually using this at this juncture to create structural images of the brain. We want functional images of the brain to tell us about how the aggregation of labeled neurons within the brain, their activity is changing. And I'll run through this just once again. So we have an array of emitter pixels, and we can, with cellular resolution, pattern light into this region that we're interrogating, 2 picosecond scale pulses. The fluorescence happens on a few nanosecond scale, and we collect all those fluorescing neurons, and we get photon counts at these independent points. And then what we have to do is demix mathematically the photon counts that are a function of the illumination, the structured illumination field, into what we really run at the end of the day, which is time records of the fluorescence for individual neurons. These happening simultaneous, the activity simultaneous in the volumes that we're interrogating. Let me tell you a little bit more about the paradigm for this lensless nanoprobe-based functional imaging. So here's our photonic nanoprobe. It has a spatially structured illumination field, and it has a spatially structured angular region of acceptance for the collection of fluorescing photons. And we'll call the overlap illumination collection fields. This is going to be likened basically to voxels in conventional recording. And with a multiplicity of these emitter and detector pixels, we end up having a multiplicity of these illumination collection fields. These are non-orthogonal. Again, you can think of them, you can liken them to point spread functions or optical transfer functions for conventional imaging, but the voxels here are non-orthogonal. Let me compare. A camera works by a flash bulb lights the room. Okay, I'm dating myself here. A flash lights the room, and then you end up having a CCD at the back end that the photons are delivered with the point spread function. It's determined by the area function for that lens. Okay, so we have a bunch of discrete pixels, but a single illumination field. Scan probe or scanned imaging, like two-photon imaging or confocal microscopy, works by broadband collection, sort of the inverse, but scanning the point around. So we're doing both. We have discrete illumination fields and discrete collection fields. And I'm going to skip this part here. We have a detailed comparison. But here's the overarching question. Obviously, building this technology is going to be expensive, a time sink, and complex. So how crude can we make these illumination collection fields to achieve spatial separation of the spiking of the neurons, complete source separation, while yet making the probe as simple, it's not simple, but as simple as possible, okay? But no simpler. So we're always guided by physical models and computational models, and at the same time as doing the heavy lifting in the nanofabrication labs to build these devices and to test them out. And given the expense in building these things and the time scale that's involved, we embarked in the first phase of our effort in building a detailed computational model for understanding how these photonic probes actually work and what their performance would be. And so I want to just run you through that very quickly now. So this is at the upper right-hand, dyslexic, upper left-hand part of the screen. You see 24 shanks, which are represented down there in the bottom part, that are implanted into brain tissue. I'm thinking of a cortical column with 100,000 neurons in a millimeter cube. And we're thinking about increasing the labeling density for these reporters from sparse, 5% of the neurons will be reporting their activity, ultimately up to 100%, and just trying to give you a representation of what that looks like. Here's what, between a pair of, or a set of 4 of these probes, this is what our volume looks like for the interrogation. For using existing reporters for calcium activity, that's for aficionados, just to describe what we're doing. We're going to use 1-photon processes and excite them at 485 nanometers and record their activity at 515, which will bias us for the calcium-rich state, indicative of the neuron actually firing. Okay? And so here's a neuron. This is roughly to scale. It fluoresces, and we collect those photons. And we can do this since the speed of our photonics is basically picosecond scale for the excitation, nanosecond scale for the decay of the chromophores. Our single photon avalanche diodes have picosecond scale timing accuracy. They're devised for fluorescence lifetime imaging, in fact. We're able to do this interrogation of this neuron somewhere between 100,000 and 500,000 times with structural illumination in between every action potential. Okay? So on the millisecond time scale, we can do these interrogations upwards of almost 1 million times. This gives us a great complexity of the measurement space for doing this demixing that I'm talking about. If we go through the numbers with a specific geometry, we find that the originally proposed architecture that I show here in the picture on the right wasn't really that great. I mean, it's great, but it's not that great. It's not great enough to do what we proposed and are funded to do. This is a little bit complicated, but these show sort of the process of demixing for increasing labeling density of these neurons. And on the x-axis, basically, is the signal amplitude or signal-to-noise ratio going from 1 to 10. Those are what the 3 blue lines are. And on the right, basically, it's the separability angle. And the way of thinking about that is that let's focus on one neuron's activity, and let's move to a hyperspace where the activity of all the other neurons is in this other part of the hyperplane. And if we can orchestrate an architecture and a illumination protocol that orthogonalizes the activity of that one neuron from the background activity of all the other neurons, then it will be fully separable, and that would be a demixing angle of 90 degrees, or cosine theta of 1 to the 0. And so that basically shows you that for 5% labeling, we're good. But as we go more and more labeling with this particular architecture, you can see more and more of the neuron distribution is falling below these lines. In other words, they're not separable. What's going on? Well, more and more neurons, this is the same thing that happens for aficionados with calcium imaging, with free space, spatial light modulators or what have you. More and more activity means more and more background fluorescence that you can't separate out from the stuff that you really want to look at. Well, we can change our architectures and search. We haven't done a Bayesian or a deterministic search yet. We've just done this sort of by the seat of our pants. But we can actually find architectures now. We've been doing this multiple times. We can find architectures where this gets better and better. And it turns out that what's needed is more spatial diversity. It's kind of a no-brainer. If you make, obviously, those illumination collection fields small enough where only one neuron is inside, you're done. But we'd like to make them as sloppy as possible, and so that's why this optimization process and this search in architecture space is important. Just to summarize this part, we started off with the purple trace on the right, which shows that as you go to increasing labeling density, so many neurons are lighting up that in fact even the 5% or the 10% that originally you could see now are no longer resolvable, to an optimized geometry where we're now up to 84%, 84,000 neurons that could be interrogated simultaneously and demixed. This is only using linear separation theory. There's so much more information in photon correlations relating to the spatial extent. Some of you know about Hanbury-Brown twist experiments and actually getting the angular subtended, sorry, I'm a little ill today, for stellar diameters. And we can do the same thing with neurons here through photon correlations. We absolutely plan on doing this, but it's not the first thing that we proposed. Okay. And here's what I mean by spatial diversity. For example, you know that you can make phased array antennas. We can make phased array emitters on our chip. We've done that using photonic delay lines. Again, these are all nanoscale components that can be made on these implantable shanks with our... I said there was no sound, but apparently there is sound on this. This is a little experiment down here in the bottom, just showing that we're using one of these photonic probes and showing that we can actually steer the beam around. These are the guys that did it. Sorry for that imposition. Okay, I'll say a few more words about the hardware, show you the progress that we've made, and then conclude. So let me show you some of the E-pixels and D-pixels. I told you about reporters, and I said that the genetically induced ones are really the most prevalent. This is a class of molecules that are found in fungi, I believe, that are light sensitive. You take those genes, you transfect them into neurons. The protein-making machinery of the neuron starts making these, and they're transported to the cell membrane, and lo and behold, now you have light-sensitive neurons, okay, even though the brain, as I said, is not light-sensitive. In the early days, typically the way people applied the light to actually stimulate the neurons to fire was implanting an optical fiber, but that delivers a bolus of light, not a physiologically useful stimulation pattern. It turns out that we can do much better. We can deliver cellular scale illumination using these photonic probes, and we can do this with a multiplicity of independently controlled microscale emitters. And to achieve this, we use a technique that comes from telecommunications called wavelength division multiplexing, and that is basically taking the absorption bandwidth for these molecular effectors. This is channel rhodopsin 2, the blue trace here. It's about a 50 nanometer wide part of the spectrum where light of any wavelength within that 50 nanometer band if you hit these channels, they'll open up and cause the cell to spike. Well, what we can do is essentially channelize our stimulation by making a whole bunch of different wavelength channels, and then delivering these wavelengths, specific wavelength channels to different points, different specific points on our probes. And this allows us to get around the multiplexing problem, which is, we can't for every neuron put in a single optical fiber. We have to figure out ways, just like I was talking about in recording, to multiplex for the stimulation. And so let me show you the progress that we've made on that. So here's the paradigm, basically. We come in with a multispectral signal in an optical fiber. We have wavelength selective elements that pick out the particular wavelength that's encoded with a particular temporal information that we want for a particular illumination pixel. And we do this all the way down the pipeline. And then those specific encoded waveforms, temporal waveforms or light signals, are then delivered to specific points. So we use wavelength division demultiplexing to spatially address specific regions. So this is an example of the power of integrated photonics. Trillions of dollars have been spent on this for telecommunication. We prototyped these probes at the Kavli Nanoscience Institute. Here are the different emitter pixels that we've demonstrated. And what's remarkable is that the light beams are extremely collimated, even though brain tissue is highly scattering. And this caught us a little off guard because I don't think this is widely appreciated or understood, let's say appreciated, generally in neuroscience, that light beams can be very well collimated in brain tissue, at least over distances that are a few times the Mie scattering length. And in this case, it means distances that are up to 500 microns or so, which is the distance between our individual pillars, if you will, that bear these individual elements. We can still keep a beam that is collimated to something like the dimensions of an individual neural soma. We've taken this technology, and our first application was essentially to use one e-pixel to stimulate part of mouse cortex while simultaneously using 2-photon free space microscopes to interrogate. This is again shallow, right? So we can use free space 2-photon microscopy and calcium imaging to understand the response of firing of neurons to this local signal. This shows a region where the green is the base fluorescence. After the stimulation, it's this yellow, and the overlay shows both. And you can see that only one neuron was lit up by this randomly inserted probe that apparently was exciting some of the neural processes that caused then backpropagation of potentials to cause this one neuron to fire. This is what it looks like after multiple trials, that one neuron fires. And then when you do broadband illumination or wide area illumination through the microscope objective, they all fire. So we know that in fact they're all properly labeled and could fire if they were stimulated optically. And I'll finally end up with this e-pixel story by showing our latest progress on massively multiplexed and addressing these individual e-pixels with fast addressing. Okay. And then the last part of the technology is developing the single photon counting detectors. My colleague at Columbia, Ken Shepard, has been working for more than a decade in developing CMOS compatible or CMOS-based single photon avalanche diode detectors with picosecond scale temporal resolution. And we've employed these directly. For this technology. I won't go into how single photon avalanche diodes work or so on. Basically, think of them as Geiger counter for photons. We're in a photon-starved regime. There are very few photons, and the cross-sectional area of capture is small enough that we need to be in a single photon regime. And we've done our first pass of developing these by using the IBM foundries, now global foundries. We've shifted this since to Taiwan. and then post-processes these into individual arrays of implantable probes at the Kavli Nanoscience Institute at Caltech. So a few more words before I conclude. So I sort of tried to throw out, it was facile and simplistic, but I tried to throw out that brain activity from 100 billion neurons at 1 hertz doesn't seem sufficient to explain how brains compute, or is unlikely to. And this is more widely beginning to be appreciated, I would say, especially in the wake of the Brain Initiative, where the first thing we proposed to do was actually to measure as many spiking neurons as possible. But a bunch of wise people have said interesting things about this. Stuart Firestein, in his great book, Ignorance, How It Drives Science, says that spikes are regarded as the language of the brain for obvious reasons. That's how they seem to talk to each other. And they're a fundamental unit of neurobiology, but we've been making all sorts of grand theories for 75 years, and some of it's true, but we haven't been focusing on much other than that, at least en masse. There are many other signals in the brain, and maybe they're perhaps the more salient features of brain activities, but meanwhile, we've been mesmerized by spikes. We should also look at what spikes don't tell us about the brain. And I threw out this idea of different multidimensional physical fields, if you will, in the brain. And if we could measure them, their spatiotemporal evolution, with sufficient resolution, we could learn a lot more about what's going on in the brain. This theme has really been nicely underscored by Corey Bargman and Eve Marder, who study respectively C. elegans, a little worm with 300 neurons, and the stomatogastric ganglion of the crab, which sort of grinds its food up, with about 30 neurons. And they point out a couple of important salient points. They say, first of all, that a circuit's function may not sufficiently be, it's not specified only by its topology. Circuit function can change entirely through neuromodulation. Neuromodulation could be more typically done by chemicals, neuromodulators, but it also could be done thermally. So the connectivity diagram only establishes potential circuit configurations, but the precise nature of the modulatory state is determined by, again, these multiphysical parameters, not just the spikings or the topology. So functional connections that give rise to a specific circuit output are configured by the neuromodulatory environment. We should be measuring that. Now, the great thing about these photonic circuits is that, that I described, is that they use local reporters to transduce into the optical domain. Right now we're looking at calcium, which is a proxy for the spiking of the neurons. A next generation of voltage reporters is coming, and great progress has been made on that. But there are many other things that we can look at. We can look at local forces, we can look at local chemicals, we can look at local temperature. All of these things should be done. Just to give you a slight glimpse at where the technology is going, this will be very brief. We can do local neuromodulator sensing, small molecule sensing using electrochemistry. If we can make these small probes, we can actually functionalize them to do amperometric measurements, and we've done that. We have prototypes and work that we've done at Caltech, which has been now spawned into wafer scale production in Grenoble. And then for larger molecules, for proteins, it's necessary to take a different path of interrogation. And here we've used microfluidics on shanks with cellular scale, sort of, if you will, ports to actually do local dialysis and to capture the protein signals, markers. I'll skip this. So let me conclude. Feynman gave this wonderful talk in 1959 at Caltech called Plenty of Room at the Bottom. And he said something that probably is anathema to a biologist, a bit arrogant. He's a physicist, okay, so. It's very easy to answer many of these things. You just look at the thing. Okay, well, he's a theorist, I'm an experimentalist. I have to build the eyeballs to look at the thing. And this can take a long time and a lot of money. In the case of LIGO, it took 50 years before, somebody was sitting September 14th and saw the evidence for two black holes colliding. My boss, Fiona Harrison at Caltech, spent the better part of 20 years developing an X-ray telescope, New Star, that gives us unprecedented vision into violent events in our galaxies. And one of my favorites is something that has direct bearing on what I talked about today. It's only 10 to the 18 times larger in volume, IceCube. I would say that this basically already proves our paradigm, and let me show you why. IceCube is a neutrino detecting array located at the South Pole, so the world's inverted here for your benefit. At the surface is the lab, IceCube lab, there it is. And going down 3 kilometers are these boreholes in which these single photon detectors have been placed. The core, and there's the Eiffel Tower for scale, the core of IceCube is this right here, the array of 86 strings, 60 sensors each, 5,160 single photon sensors. That sounds like about the number we have, right? Here's what the single photon sensors look like. They're not microscopic, but in fact, they're things like that. And they go down a borehole, and then they're frozen in. You backfill it with water, and you have your array. And we can do the same thing for the brain at a fraction of the cost. But the more serious point is that this works because it follows a paradigm that's completely unfamiliar to the neuroscience community, and that is of people working in a very coherent, front-to-back way, building subsystems that are designed to be interoperable, that are cohered by one sort of central agency. In this case, it was the University of Wisconsin-Madison that managed the IceCube project and allowed for a complex system to be built to measure complex stuff. And a follow-on thing that the original 6 proposers of the Brain Initiative wrote last year or so was that we still need to do this in the Brain Initiative. We still need to think about this large-scale science, building an observatory for the brain with this advanced technology. I'll conclude with the following comments. I mentioned a new paradigm for doing deep brain interrogation. It's wonderful what can be done with free space functional optical imaging, but it's not going to get us there for mouse brains, and it's a non-starter for the human brain. I proposed actually implanting all of the elements of an imaging system into the brain on length scales that are comparable to the optical attenuation length. And the beauty of this paradigm is that we can mass produce these systems using silicon integrated nanophotonics technology and deliver them worldwide to users. For roadmaps, I talked about scaling up electrophysiology in the next 3 years to 100,000 channels. It's a longer quest to do these photonic probes. I think in the next 2 or 3 years we'll have 1 photon functional imaging systems, but really the sky is the limit when we actually transfer this, when we actually understand how to do sub-picosecond interrogation using integrated nanophotonics. Dispersion control on chips is necessary. A new generation of 2 photon functional imaging will become possible, and lensless structural imaging as well will be possible. Let me just skip this. And that's my talk. I just want to acknowledge all the wonderful people that I have the privilege of working with. Thank you.